Within Thermo Fisher Scientific, we're working very, very closely with our customers within the bioanalytical field to understand their issues and how we can address those issues, and that's leading us to develop new innovative technologies that really create solutions to address those major pain points that they have. With the advent of high potency drugs, it means that we have compounds or drug candidates that are beneficial to our customers but are much lower concentrations. So as a result of that, our quantitation levels and our sensitivity levels are being pushed further and further down. What's happening is that we're getting to a point where we're getting much lower sample volumes. So we have to consider that that is something that our technologies need to be able to address. And in addition to that, there's obviously the ever-evolving compliance to regulations, the changing face of regulations, particularly around reproducibility, where our solutions have to conform to and aid our customers in addressing those needs. Developing new technologies aids our customers developing new workflows, improve their efficiency of the workflows, improve the quality of their results, which is fundamental. So we've been developing some new technologies with our partners in the line of sample preparation. So we've brought online new next generation sample preparation technologies, such as the solar microelution product. So that's got considerable benefit at a key stage in the workflow. What the benefits are there is that we have a very robust, reproducible format, but it also allows us to have a big pre-concentration set. So that means that we get a big increase in our sensitivity. It's also got the ability to process low sample volumes. We can extract in very low sample volumes. So that means that we can remove the blowdown step, which means we have a more efficient process, and also has got a benefit for the biopharmaceutical industry when we're looking at those protein and peptides that are susceptible to those non-specific binding issues and solvation issues. Removing that step really has benefit there. But it's not just obviously in sample preparation, it's all at different points throughout the entire workflow. So we're also looking at our collection devices, so our range of collection plates. What we have there are collection plates that are made of high purity materials. They don't leach any components into our sample. That means that we don't get any additional suppression of our signal within our mass spectrometry detection. Also, the sample integrity is ensured as well because we have the ability to seal the plate with a siliconized mat. So that means that we don't get evaporation of our samples. And then we move on to the next stage of the analysis, where we're developing new next generation UHPLC column technologies and systems, such as the AccuCore Vanquish columns and the Vanquish UHPLC system, which are able to deliver more efficient chromatography, better peak shape, more accurate quantitation, but also to really push forward and deliver higher throughput to a customer. And that's then coupled obviously with state-of-the-art mass spectrometry solutions such as triple quad technology in terms of the Endura, the Quantiva, and high resolution mass spectrometry systems as well, in order to get that real high level of quantitation and qualification accuracy that we really need within this field. And that also feeds into how we're processing our samples. So looking at the Chromeleon software, we're taking away some of the pain points there, making it very easy and accurate to interpret and qualify our results, feeding into the wider LIMS system, the Watson system, which is globally world-renowned within the field, which is helping to bring to the fore workflow solutions, sample integrity, sample tracking for GLP analysis. So you can see that really each individual section has its own merits and its own benefits to the workflow, but overall, all of those solutions together are a much more powerful offering and really trying to, I suppose, change the dynamics and within quantitative bioanalysis. The customers are really impressed. I think one of the main things that they're impressed with is our commitment to work with them, to really understand what their pain points are. There's no point bringing a technology to market that really doesn't address those issues. So it's working with them closely, understanding what are the issues that they're facing now and in the future, and how we can develop those technologies to facilitate those needs and bring benefit to their workflow. And I think they really feel the benefit of working with us in that way, and obviously we do as well, because we're getting access to understanding how the dynamic in the marketplace is moving forward and bringing the appropriate and the correct workflow solutions to aid in benefiting those customers.